Thank you for joining me tonight, Simak. How are you doing?
Very well. Thank you for the invitation and chance to talk about our work.
No worries. Thank you for joining us. I think a good place to start would be just about your background. How did you get into breast cancer research?
Right. So I actually started at school with interest in science. I went in, did a first degree at Imperial College London, studying biochemistry and molecular biology, which was still quite a new field, really. And then from there, I went to the Roslin Institute in Edinburgh to do my PhD. And I was, in fact, working on mammary gland biology in sheep, of all things. And so that led to the interest in how genes work, how they're controlled. Mammary gland in the sheep to the breast obviously was a straightforward move in that context, where I went to do my postdoc with my supervisor, Professor Pierre Chaumont in France, who had in fact discovered or cloned the estrogen receptor gene. And because of its importance in breast cancer, it was then an obvious step to try to work on breast cancer and find out how the estrogen receptor works and why it's so important in breast cancer development and progression.
Okay, so your project with us is looking at secondary breast cancer, and your background is mainly focused on the ER gene, isn't it? Am I correct?
Yeah.
So perhaps we should start a bit with what exactly is secondary breast cancer?
Well, I guess from my point of view as a scientist, it would be breast cancer which has progressed. So we have what we call primary breast cancer, which would be localized disease. So it would be a tumor in the breast, and when that progresses to become more metastatic, perhaps moving to other sites in the body, that's what I would refer to as secondary or metastatic breast cancer.
Okay, and within that realm, what is so significant about the ER gene? In fact, actually, perhaps we should start with what is the ER gene?
So the hormone estrogen is really an important driver of breast cancer development as well as its progression. And how the estrogen works is by binding to a protein known as the estrogen receptor. And the estrogen receptor protein is then switched on when it's bound by estrogen, and is able to drive expression of hundreds and hundreds of genes, which together essentially push or allow the cells to proliferate. And so that's obviously a normal process in the breast and in other tissues where the estrogen receptor signaling is required. But in breast cancer, in 70 to 80% of breast cancers, what we find is that there are high levels of the estrogen receptor protein. And so you've got hyperactive signaling. So you're getting really high levels of expression of of the genes that it controls. And that's what pushes uncontrolled growth of the breast cells, making them cancerous and eventually also spreading to other tissues.
And how is it involved in cancer spreading around the body? Is it that kind of uncontrolled proliferated growth, or is it also other mechanisms that it's linked to?
So the estrogen receptor, as I said, drives gene expression. So it's what we call a transcription factor. So it binds to sequences in genes, and in doing so, it is able to cause those genes to be expressed. Those genes will then make proteins which will tell the cell to replicate the DNA, and also then to divide, and so you increase the number of cells. And in doing this, you essentially end up with this uncontrolled growth, essentially because the estrogen receptor is controlling the levels of these genes, and by having a lot more of the estrogen receptor protein, you lose a lot of the normal constraints on estrogen receptor activity. So you're essentially getting much more activity than you would get in a normal cell setting.
Okay. And with the ER gene, what are kind of the main challenges when it comes to studying this?
Right. So because we know, and we've now known for a very long time, that estrogen promotes development of breast cancer. And so certainly even more than 100 years ago, some patients were treated by removing the ovaries. And that was before estrogen had actually been identified. So we've known for a long time that estrogen, working through the estrogen receptor, is so important in breast cancer. And that's led to the really, really good treatments. And they fall into two main classes. The first are what are called aromatase inhibitors. And what they do is to prevent the synthesis of estrogen in the body. And so no estrogen receptor is off. So it can't then control the expression of those genes that drive growth of the tumor. The second class of drugs are ones that bind to the estrogen receptor in place of the estrogen. And their binding, rather than activating the receptor, in fact represses it and may even cause degradation of the protein. So there is no estrogen receptor in those cells, and so the cells stop growing. So those are the two main treatments. And, they have been extremely important in management of breast cancer following surgical removal of the tumor. So in terms of what we now know, these are relatively recent discoveries, as probably a lot of the viewers will know. Cancers evolve, they develop mutations in many genes that can help the tumor to continue to proliferate or spread to other tissues. And one of these recent findings is the fact that to overcome the block that's provided, certainly by these estrogen biosynthesis inhibitors, the aromatase inhibitors, cancer cells develop mutations in the estrogen receptor gene, which means that you are making estrogen receptor protein, which actually doesn't require estrogen to be activated. And so those drugs that would block estrogen biosynthesis are no longer working because the estrogen receptor is what we call constitutively active.
Okay, and so you're looking at these changes in the estrogen receptor. How do you do that? What is the process of looking at these genetic changes?
Yes, so now quite a large number of different mutations in the estrogen receptor gene have been identified. And what we are looking to do is to work out how those mutations work. How is it that they make the receptor active even in the absence of the estrogens? And so the way that we're doing it, and again, many of the viewers will know about the CRISPR technology. So this is a method by which you can make changes in the DNA, and there are lots of possible therapeutic outcomes from that. For example, treating many diseases like sickle cell anemia and so on. And so we're using that CRISPR approach to take breast cancer cells, introduce these mutations into the estrogen receptor gene in the breast cancer cells. Because at the moment, there are very few models. There are no breast cancer cell lines that we can work on in the laboratory where these mutations are already present. So we've had to introduce them, and we've used the CRISPR methodology to do this. And so what we've done is to make breast cancer cell models with a large number of these different mutations that are found. And then we are using the kind of technologies that have emerged from sequencing of the human genome to find out all of the genes that these mutant estrogen receptors are regulating, and try and identify essentially additional vulnerabilities in those cancer cells that could be targeted when a patient develops secondary breast cancer where the estrogen receptor gene is mutated.
And so is there possibility in the future, would it be you would look at these mutations and then hopefully find a new treatment that could target those mutations?
Absolutely. So we're using these sort of deep sequencing approaches, as they're called, where we can look at expression of every single gene in these cells. We can compare them to the breast cancer cells which don't have a mutated estrogen receptor, look at the differences and see whether that allows us to identify particular genes that could be new targets for therapy.
And I'm guessing a lot of these drugs wouldn't exist yet. Like how do you go through the process of, so you identify these mutations, you're looking for a treatment. How do you find that treatment? I can imagine that's quite a complicated process.
Yeah, so once we identify the gene, what we would do is to manipulate it in those cancer cells to determine if that gene is really important for the growth of the mutant cells. And then if it is, we would try and identify ways of targeting it using drugs. Ideally, we would identify targets for which there are already cancer drugs, and that's certainly a possibility. But if there aren't, then there would be the requirement for developing new drugs. And we've done this before. So we have a drug that we've developed, which was essentially developed to be able to target the estrogen receptor and block its activity when the cells had become resistant. And it was an enzyme called, protein called CDK7. And we've developed a drug, taken it all the way into clinical trials, and we've just reported recently on the fact that in estrogen receptor positive breast cancer, many patients do show a clinical benefit. These are still very early days, but we do have the potential to use the information we get from these models to either use cancer drugs that already exist, which would of course be a much faster route to translation into helping treating patients. But if not, then to develop new therapies.
And I know this might be quite a complicated answer, but how do you develop those new therapies? Is that a complicated path?
Yeah, it takes a long time, and there are a number of reasons for that. Firstly, once we have identified a protein, a target that we want a drug against, then you need to have develop a biochemical assay generally to be able to demonstrate that any small molecules, any chemicals that you develop to target that protein, in fact, do block its activity. From there, you have to demonstrate that you can move that into a situation where it becomes a drug. That means it can be administered to patients safely, and that is a major problem. You don't want to be having toxicity in patients because of the drug that you're administering. So there are lots of tests that we need to do in developing that. And actually that part, going from finding a chemical that is able to, that has the biological activity that we want, to something that is safe and also easy to administer. So can be, for example, ideally taken in tablet form by patients, and has no side effects. That's the part that takes a long time. So in developing our CDK7 inhibitor, for example, I think we got to a very good drug in about 6 months, or chemical in about 6 months, but then found that, in fact, it works well in a test tube, but when we treat breast cancer cells with it, it doesn't get into the cells. So it was dead. And then we spent another 9 years in going from that to something that patients could take as a tablet. And as I say, now that's going into so it's gone through the safety aspects in patients, or what's called a phase 1 clinical trial, and it's now gone into a phase 2 clinical trial where it will be matched head to head with another breast cancer therapy, and that will be in patients with secondary breast cancer.
Yeah,
and as it turns out, by the way, the other point I did want to make is that we got this target because of what we wanted to achieve in breast cancer. It turns out that this particular enzyme and this particular drug will in fact be of benefit in many other cancer types. So we're exploring prostate cancer, for example, but also other cancer types are being evaluated now in these new clinical trials.
Yeah, that tends to be the case, doesn't it? That sometimes there's overlap between different types of cancer. And yeah, and so perhaps we should talk a bit about clinical trials as well. Like, so there's stage 1, 2, and then 3 and 4. So yeah, like you said, it can be a long process for these projects of going from bench side to it actually being used to treat cancer. But yeah, you've mentioned a bit about the progress you made so far, but what have you found anything interesting so far? Any developments?
Well, we certainly know already, in terms of possible treatments, that the drug that we've developed it is effective in patients with estrogen receptor mutations. So that's certainly very good finding. In terms of what we're hoping to achieve with the recent wonderful funding from Breast Cancer Now, looking at these mutations, is to be able to see identified drug targets that would be directly applicable. So one of the things we've found is that these mutations in the estrogen receptor do make the cancer insensitive to aromatase inhibitors. But there is still sensitivity to those what are called anti-estrogen, so the drugs that prevent the estrogen from binding to the estrogen receptor and can cause degradation or destruction of the estrogen receptor protein.
Okay.
And we're finding that these mutations are actually still sensitive to some of those anti-estrogenic drugs.
And so is your hope, ideally, with this research, that the drug would be used to treat people with secondary breast cancer?
Absolutely, yeah.
Okay. Yeah, I mean, yeah, that's fantastic, because I know that's an area that lacks treatment, and we really need treatments in that area. So what is the ultimate goal with this research? Is it that you want to find a drug that will go to clinical trials and be effective? Is that kind of the high hopes you have for this research?
Yes, that's really where we want to end up, is at least be in a position to have these targets which could then be used as the basis for either discovering new drugs or, as I keep coming back to, what's called repurposing other treatments, because that would be a much quicker route to helping patients.
And where else do you kind of see this research going? What other? Because I know you mentioned the ER gene is involved in a lot of different areas. Is there any other area you'd like to investigate the ER gene?
We certainly the estrogen receptor is important in the context of cancer, in endometrial cancer. And you again do see mutations in the estrogen receptor gene in some patients with endometrial cancer. We are also, because the estrogen receptor as a driver of gene expression turns out to be related quite closely to the androgen receptor, which the androgen receptor does exactly the same job in prostate cancer. So it drives expression of genes which allow the prostate cancers to grow. And so we do believe that a lot of the kind of information we get on estrogen receptor mutations and therapies actually would have some crossover also to other cancer types, particularly endometrial cancer, and in the case of the androgen receptor, which also have you see a lot of mutations in the androgen receptor gene as well. So, it's our primary aim is in the context of helping breast cancer patients, but I think in terms of understanding biology of cancer, there are ramifications outside just estrogen receptor positive breast cancer for sure.
That's brilliant. So so some of the findings from this project could be applicable to other types of cancer, and
most definitely, yeah.
Also, brilliant. Awesome. Really, really interesting stuff, and really excited to see the results you come. Hopefully it will be a repurpose and not a new drug, because like you said, I'm sure that would be much easier. Brilliant. Well, thank you so much for giving up your some of your evening to talk to us. That was really fascinating. And I hope everyone who tuned in found it as interesting as I did. You can please make sure to visit our website. You can learn more about CMAC's research and also the other research we're funding, as well as services that we offer. And I'll see you all next time, where we'll be chatting with more breast cancer now researchers about their work. Again, CMAC, thank you so much for talking to us tonight.
Thank you.